Okay. 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 Okay.
But then to try and give us about 5 minutes of questions. Don't worry, I won't come chasing after you until we get to 19 minutes and 50 seconds.
I will try to finish in time.
Okay, so our next talk is by Vedrana Vidulin from the Joseph Stefan Institute in Slovenia, and she's going to talk to us about the evolutionary signal in metagenome phyletic profiles predicts many gene functions.
Hi, my name is Vedrana Vidulin, and today I will present you results of a study performed in cooperation with Fran Supek, Tomislav Šmuc, and Sasha Jarosky. Increasing availability of metagenomic data provides us an opportunity to use this data for automatic function prediction. Currently, many of automatic function prediction methods exploit genomic data for constructing classification models. However, there are far more metagenomes available in public databases than genomes, and metagenomic databases also tend to grow faster. So, motivated by the availability of metagenomic data, we constructed metagenome phyletic profiles and evaluated its predictive performance in the context of automatic function prediction. MPP encodes metagenomic data in a representation that consists of relative abundances of gene families across metagenomes. Gene families are then annotated with geoterms from the UniProt-GOAT database and are propagated using gene ontology to obtain a hierarchical class in the form of a directed acyclic graph. Then we employed CLUS HMC random forests machine learning algorithm on the MPP dataset to obtain a classification model. We used class HMC. HMC, by the way, stands for hierarchical multi-label classification, because it is able to use an information from the hierarchical class during the model construction phase in order to improve model's predictive accuracy. We evaluated MPP in several steps. First, we were interested in whether we can predict a broad range of gene functions using MPP. Then we constructed multiple MPPs, each with metagenomes from different environments, and we measured a degree of overlap in gene functions they predict. We also compared MPPs with phyletic profiles, a similar representation that is based on genomic data. Then we took 16S rRNA data, constructed MPP from it, and analyzed MPP's predictive accuracy. And finally, we performed a simulation to see how we can maximize MPP predictive accuracy. So to answer the question whether we can predict a broad range of gene functions with MPP, we constructed two MPPs. First, name MPP-H is composed of around 1,200 human gut metagenomes from the integrated reference catalog of the human gut database. The second, name MPP-O, is composed of 139 ocean metagenomes from the Tara Oceans database. So for the two MPPs, we first count a number of learnable gene functions. By learnable, we mean that a function is predicted at least once by MPP at the precision threshold of 50%. So in total, there are around 450 learnable geofunctions in the case of MPP-H, and around 320 in the case of MPP-O. We also measured predictive accuracy for each geofunction. Predictive accuracy is here and area under precision recall curve, and observed the distributions of accuracies. So we can see the similar trend for both MPPs. MPP is a bit better in predicting specific and general geofunctions than medium-specific that are typically found in the middle of gene ontology. And we also compared MPP's predictive accuracy against random baseline. And we can also see that it performs significantly better than the baseline. When we compared MPP-H and MPP-O, we found cases where one is considerably better at predicting a certain function than another. So, for example, in the case of function metagenesis, MPP-H predicts it considerably better than MPP-O. This case is meaningful because metanogens are an essential part of the human gut, promoting fermentation of carbohydrates, while metanogens are not highly represented in oceans. Another example is the function photosynthetic membrane, which is well learned by MPP-O and poorly learned by MPP-H. This also is a meaningful case because in oceans we have cyanobacteria that use photosynthesis while there is no light in a human gut to promote photosynthesis. So motivated by these individual cases, we decided to measure a degree of overlap between two MPPs in terms of gene functions they predict. So at the very stringent threshold of the precision of 90%, we can see that only 35% of gene functions are simultaneously learnable by both MPPs, which indicates high complementarity among MPPs that represent different environments. Next, we wanted to see whether we can see the similarly high level of complementarity with more than two environments. So we collected metagenomes from 7 environments like freshwater, ocean, soil, human associated metagenomes, and so on. We constructed MPP for each environment, and then we measured complementarity as a proportion of gene functions that are simultaneously learnable by different number of MPPs. So at the precision level of 50%, we can see that similar proportion of gene functions is learnable solely by a single MPP and simultaneously by all 7 MPPs. Detailed analysis showed that those functions that are learnable by all 7 MPPs, the most of them are related to housekeeping genes like DNA repair and translation. While on the other side, we can see that almost half of the gene functions is learnable by less than half of the MPPs, which confirms relatively high complementarity among MPPs representing different environments. Next, we compared MPP with phyletic profiles. So phyletic profiles are a representation which encodes presence and absence patterns of gene families across genomes. To compare the two, we used 3 instances of MPPs, MPP-H, MPP-O, and we also constructed MPP-I, which is composed of metagenomes from the 7 environments presented on the previous slide. And we further constructed matched phyletic profiles. We matched them in a manner to include in phyletic profiles only genomes of those organisms belonging to phyla represented within environment of the MPP. So the results showed that when we have MPP with metagenomes only from a single environment, then phyletic profiles can learn many functions that are not accessible to MPPs and in a manner outperform MPPs. However, if we include metagenomes from a diverse environment, like is the case with MPP-I, then the performance between the two is similar. Approximately the similar proportion of functions is learnable by MPP that is not accessible to phyletic profiles and vice versa. 16S rRNA sequencing is cheaper than a whole metagenome sequencing. Consequently, there are far more 16S samples available than whole metagenomes. So we collected 20,000 16S rRNA samples representing 64 environments from the CHITA database. Then we predicted gene content from those samples using PyCrust. PyCrust uses a database with whole genomes of cultured organisms to predict gene content. So from the predicted gene content, we constructed MPP16S, and we compared it against MPPI, which uses metagenomes that are obtained by whole genome metagenome sequencing. We first compared overall predictive accuracy of MPPI and MPP16S, and we were surprised to see that MPP16S performs a bit better. So we hypothesized that in the case of rare gene families, by using PyCrust, we were able to more correctly predict abundances of rare genes. That is the case with whole metagenome sequencing with low resolution. So to test this hypothesis, we divided gene families into rare and abundant gene families, and for each of them we constructed a separate MPP. So the results show that in the case of abundant gene families, MPPI with whole metagenomes did perform better. But when in the case of rare gene families, the advantage of MPPI diminished, which confirmed our hypothesis. Finally, we performed a simulation to understand how can we maximize predictive accuracy of MPP. So we gradually added metagenomes to MPP and measure predictive accuracy at different points. We used two sampling strategies. First is maximal diversity sampling strategy, where we edit metagenomes in a manner to keep the same, approximately same ratio of metagenomes from different environments. And another is minimal diversity sampling, in which we first edit metagenomes from the largest environment, then from the second largest, and so on. So the simulation showed that a better strategy is to use maximal diversity sampling. So if we want to increase predictive accuracy of MPP, it's better to add metagenomes from diverse environments than just to add more metagenomes from a single environment. So if you find MPPs interesting for your research, you can read about them in more details in our paper published in journal Microbiome. And we also published MPP datasets together with the datasets from our previous automatic function prediction study. And if you want to talk about MPPs, you can find me at the poster station C14. So thank you for your attention.
Any questions?
When you used PyCrust to expand the 16S data sets,
Yes.
our experience with that code is that it works better for human-associated, human-associated metagenomic experiments,
Okay.
and 16X experiments. So I was wondering if maybe you could see if your performance improvement in that right-hand plot on just
sorry, I
the...
didn't hear you.
I know you break down the MPPs based on human-associated microbial ocean. And I think PyCrust might, it's just a comment, it definitely would have to be checked, but I think PyCrust works a lot better when it's human-associated because a lot of the reference genomes are there, whereas the diversity in some of those other environments probably overwhelms
You
the
mean
code a
that
little
I
bit.
tried the experiment just separated by different
If you
environments?
separate it out for just the human MPPs, maybe you would see a little bit more performance increase. But like,
Thank you for the comment.
Any other questions? Okay, I'll have a quick question then.
Okay.
So your method performed better when you had general and specific functions, but not so well when you had this kind of medium
Medium-specific,
specificity.
yes.
Why do you think that is?
I didn't look at the details in this case, so I'm not sure, actually.
Right. Any other questions? Okay, in that case, we'll thank the speaker once again. Thank you.